Okay, great. That was quite something. Thank you very much for the opportunity to tell the story today. I'll be making some forward-looking statements, and those are covered like that? Okay, great, thanks. By Safe Harbor. First of all, I'll give you a quick corporate overview. First of all, we are a clinical stage biotechnology company. We take cells from the placenta, we develop them in a proprietary bioreactor, and then we use them to treat a variety of inflammatory and ischemic conditions. Our market cap is about $250 million currently. We have about $57–58 million in the bank, no debt. We burn about $20 million a year. Our average 90-day volume is about 950,000 shares a day. About 550,000 of those shares are here on the NASDAQ. We're also traded on the Tel Aviv exchange, where the rest of them are. We have 165 employees, and we just received FDA and German regulatory approval of our newest manufacturing facility. We're based in Israel. But the facility was approved as comparable to our older facility, which shows again that we have batch-to-batch consistency and the ability to produce well-controlled clinical-grade products. Investment highlights, pretty straightforward. We have 2 completed phase 1/2 studies, both of them showing really nice results, which I'll review with you. We have our FDA-approved in-house manufacturing facility in which we use our proprietary 3-dimensional bioreactors to produce our cell products. So we really have control end-to-end from development of the cells, manufacturing ourselves and our own in-house control, and out to developing our therapeutic products. We have active programs in 4 spaces right now. I'll review those quickly in a bit as well. We have strong IP value. We have 28 patents and about 100 applications pending. And we have a very strong management team, which I will show you a bit about in a while. First of all, our active clinical programs are in 4 spaces. We're in the cardiovascular space, where we've had 1 completed phase 1 trial and an ongoing multinational phase 2 trial. Both of those, we have an out-licensing partnership with in South Korea for a portion of that work. We just announced in January phase 2 muscle injury data, which was extremely positive. And I'll go through that as well quickly. We have a phase 1 pulmonary hypertension trial in partnership with United Therapeutics, and that's been enrolling out in Australia. And we are entering now into preeclampsia, which is it's a leading killer of pregnant women in the U.S. It occurs in about 3% of pregnancies, and there's no cure except delivery of the baby. And I'll review that as well. So first of all, in terms of our collaborations, we're very proud of them because they're really showing valuation of our company that came out positively and really support for our products. The one we signed most recently was with Traub Bio and Diostech. We're very interested in Asia and working in Asia, as other people have mentioned. And what we have with them is a very attractive collaboration. We are going into a phase 2/3 in critical limb ischemia, which they will be paying for completely for all the research. This indication also fulfills orphan disease status in South Korea, which means that one can go to market after a phase 2/3 if it's a strong study. And it's a sort of fast-track pathway, which is very attractive. We are also having them join into enrollment in our ongoing phase 2 intermittent claudication trial, and they will be paying for everything trial-wise for their portion of that study. This has already been approved, and we hope to begin enrollment in the first half of this year. If these products go to market, we have a 50/50 joint venture for the profits, and they will be covering expenses of marketing and commercialization. Our other collaboration is with United Therapeutics. They're the ones running that phase 1 pulmonary hypertension trial. We got $7 million upfront payments, and we have up to $55 million worth of milestone payment potential. They're covering some of the development costs for the trials, and we are going to get single-digit royalties on growth profits if it goes to market. So let me review our clinical program. As I said, we reported top-line data on a phase 2 trial of muscle injury. This was done at the Charité Hospital in Berlin. We wanted a very focused trial that would really show how well our cells would work, so we picked something where it's only damage to one specific muscle with a very clear endpoint that's very easily measurable. So what we did is we had the trial done in hip replacement surgery. There's an approach where you repair the hip where you have to go in through the behind and you cut a muscle called the gluteus muscle. And one of the problems with this kind of surgery is that afterwards the muscle does not repair properly and the person will develop a limp and is not able to walk properly anymore. So the way that the trial was run is the surgeon would inject either our cells or placebo into this muscle after he had repaired it and before closing the skin. And what we did was what we announced was 6-month top-line data looking at 2 primary efficacy points, one primary, 1 secondary efficacy point, and as well looking at safety. So I will show you we had really, really impressive results. We did not expect anything this good. This was a 20-person trial, and we had very statistically significant findings. For our 150 million cell dose, we had 500% improvement in muscle contractile force over placebo with a p-value of 0.0067. And this is easy to measure. There's a special machine. It measures actual force of the person's muscle pressing on a lever. So it's pretty, hard and fast endpoint. And we were very, very happy with this. One of our secondary endpoints we also announced is 6 months out. We looked at muscle volume measured by MRI. And this as well, for this 150 million cell dose, we had a 316% increase in muscle volume over placebo with a p-value of 0.004. And nobody has reported this kind of significant results in a cell therapy trial, and we did not expect something this positive from a small trial like that. So we were very, very happy. We had no severe adverse events. We have several still secondary endpoints whose data is being analyzed, which we'll be reporting on later this year. And we're using this really as a springboard into the orthopedic space. Dr. Rodeo at the Hospital for Special Surgery did preclinical work for us. He just presented a poster on this tendon injury preclinical work, and we're looking to move into this type of soft tissue orthopedic indication, whether it's rotator cuff or other tendon injury, and that's really where we're going with this work. We have peripheral artery disease is an area we're looking at 2 subsets of the disease. One is intermittent claudication, and one is critical limb ischemia. These are progressions when you get clogging of the arteries to the leg by cholesterol primarily. What happens is the blood flow reduces and you get damage to the tissue. So the first subset of the disease is something called intermittent claudication, where it becomes painful to walk. Progressively more painful till it's difficult to be mobile at all. And as that progresses the disease, the next subset is CLI, where you get tissue breakdown, gangrene, and unfortunately amputation. So we had, this was our first trial in humans. We had work done at Duke, University of Alabama, Stanford, and at the Charité in Berlin, and 2 other universities were helping with the analysis. We had 27 patients. As a first in human patients, this was not a blinded, this was an open label trial for obvious reasons. And we looked at efficacy at 3 months, but in interest of time, I'll just look at our 12-month follow-up. What we had at 12 months is another hard and fast, easily measurable endpoint. Can you walk after a year? Rather, are you alive after a year, and do you have your leg? So there's no, fudging on that, pretty much. So what we saw after 1 year with treatment of our cells is we had about 85% amputation-free survival. And we don't have a control, so just for the sake of a graph, we put a historical control from published data from another company. So we essentially had a 59% reduction in risk compared to that historical control. If you don't treat with anything, you have about a 53-54% amputation-free survival at 1 year. Our other trial in this area is IC, intermittent claudication, that first one where you have trouble walking. And in this case, this is an ongoing trial in the US, Germany, initiating in Israel, and approved and ready to initiate in South Korea as well. And here as well, we have a very hard and fast endpoint, maximal walking distance at 12 months compared to maximal walking distance at baseline. So there are other outcomes, but that's the primary one. We're very focused on those in terms of regulatory considerations that have an appropriate endpoint. We have a phase 1 study in pulmonary arterial hypertension that's going on with our partners, United Therapeutics in Australia. We've begun enrolling there. There we go. The last area, our fourth program we're going into, is something called Preeclampsia. It's a leading killer of pregnant women in the U.S. and the developed world. It occurs in about 3% of pregnancies, and about a quarter of those cases of preeclampsia go into severe preeclampsia, which can lead to stroke, grand mal seizures, liver failure, and unfortunately death as well. The disease is characterized by elevated blood pressure and initially end organ damage in kidneys, or signs of damage in liver and elsewhere. There's absolutely no cure for this except delivery of the baby. The problem is, of course, that there's no drug, there's no cure, and the disease can come on at 20 weeks, which means that you might wind up having to take out a non-viable baby for the sake of saving the mother. Or if it comes on at 25, 26, 27 weeks, you may have to take out a baby that's on the really bad prognostics for long-term survival and for intact survival. And from a parent's perspective, it costs about $50,000 a week to keep a baby in these early weeks alive in the NICU, so they're also very eager to find something for that. The American Heart Association recently identified preeclampsia as an independent risk factor for heart disease and stroke in women. So women who've had preeclampsia decades later are still at a double the risk of stroke because of that exposure. We're planning to go ahead from preclinical trials to clinical trials. We also have very strong preclinical data, which we've recently announced. One trial looking at efficacy was done at Texas A&M with rodent models of preeclampsia. We had normalization of blood pressure and normalization of kidney function in 2 animal models of preeclampsia that were tested. And we also had tox screening at Charles River Laboratories, which showed no anomalies, no sign of defects either in the pregnant rodent or in the fetus that was looked at along the way, and also no evidence of the cells crossing into the fetus. And this is very important because this is one of the things we stress with our cells. They're mesenchymal-like adherent stromal cells. They do not differentiate. They do not engraft. We inject them in the muscle. They stay in the muscle. They communicate with damaged tissue. They secrete therapeutic proteins. And after about a month, they begin to wash out. So it's a very neat and clean, more like a drug really than some kind of a cell therapy in the sense of engraftment or staying in the body. We are anticipating to go into the clinic this year with a phase 1/2. We've also applied for orphan status with the FDA just recently. One of the main things we have in differentiates us from other companies is not only do we have a special technology, we control it and we do our own manufacturing. So we have really end-to-end control of the whole process, which is a big differentiator since many people are using Lonza to provide them to manufacture their cells. So first of all, there are some very fundamental advantages to the technology we have. It's extremely efficient production technology. This is a fully automated system. It avoids a lot of issues of contamination or problems with batch-to-batch consistency. It's supremely upscalable. We have in our new facility, we're able to scale up to 150,000 doses a year. And very low cost of goods, several dollars for a million cells, which is extremely low. We have, as I reported with the FDA and the German regulatory approval of our facility, we can see that we've really achieved strong batch-to-batch consistency and clear ability to manufacture cells in clinical grade and reliably. And we can by controlling the environment in these bioreactors, we can sort of push our cells into different products. So we're not a one-product company. We have one product that we're exploring now. We have several other products in the pipeline being developed. One of those is actually, a second product is actually being worked on right now at the NIH. They're expanding the testing of our cells. They're looking at them for use in acute radiation syndrome, which highlights some of the things about our cells. They're off the shelf, no tissue matching, they can be storable, and they are easily administered into the muscle. So they're really a perfect setup for this type of indication. And so we have several products coming along. The culturing efficiency, again, this is a huge deal for us. As you can see, we have with one 5-liter bioreactor, which grows the cells 3-dimensionally in scaffolding, we can make the same number of cells as 70 of these 10-well multi-trays. These multi-trays, you need hoods and people and a whole process to grow the cells. And in our case, we need computers, and that's very, very few people. We also have the ability to control, really fine-tune, precise control of the environment, and to keep the cells in an environment where they can grow optimally. Because in a 3-dimensional environment, it mimics how they grow in us, and they get a lot of characteristics that differentiate them from what can be grown in 2D. It also means that we can barcode our cells. Any cells that are grown in this type of 3D bioreactor has expressed certain genes which are not expressed if they're grown in 2D, which makes it very easy for us to protect our IP. Clinical milestones for this year, we had phase 2 muscle injury, which we achieved top-line data announcement. We had German regulatory approval of our new facility. We had FDA approval of our new facility. We achieved those 3. We're now applying and hoping to begin clinical trials of our cells in Israel for preeclampsia. We've applied, we've already received the Korean FDA's approval of our phase 2 IC trial, which they'll be joining too, so we anticipate to begin enrollment there as well this year in first half. And we're also anticipating to get approval to begin the phase 2/3 CLI trial in South Korea, again, which, if that goes well, gives us the potential to market because of the regulatory environment there. We are looking, as I said, to move into an orthopedic indication, and we're looking for some kind of early data from the phase 1 trial being run by United Therapeutics at some point next year. So basically, just to reiterate, we've got 2 very strong points of data showing efficacy in 2 separate trials. We have a proprietary 3D manufacturing technology, which is really first in class. We have our own facility, so we control the entire process end-to-end and ensure the quality and the consistency. We've gotten 2 very attractive collaborations, which are really votes of confidence in what our technology can do and the potential. We have the FDA and the German regulatory approval of our facilities. We have significant cash balances, partnerships which will provide, which can provide additional capital, and we are also actively engaged in conversations for additional partnerships. We have significant value in our IP, and we have a very, very committed management team that's had a vision for this company all along. I have a non-functional pointer. Thankfully, I've done. Oh, there they are. So that's the management team. You can see Zami Aberman is the chairman and CEO. He's got extensive history, successful track record. He was initially in high tech and moved over into biotech with his interest in our bioreactor and in the company. We have a COO, Yaki Anai, who was CFO of a division of Elbit. We have a CFO now, Boaz Gur-Levi, who was CFO of a unit at Abbott. So it's a very strong team. Our CMO is, we poached from Teva, and it's been a good time. It's a great time. It's a very exciting time for the company. Thank you very much for your attention.